All right. This is awkward knowing so many people are listening to me, but it's so quiet. All right. I am very pleased to introduce Julia McGonigle and her PhD defense today. I'll give a brief intro and then hand it over to her. Julia came to us by way of Oregon State University, where she majored in botany and conducted research on plant reproduction. She also spent a summer doing research with the NASA Student Airborne Research Program, where she researched toxic algal blooms and took a step closer to microbiology, which she had become interested in. And then, so to pursue her interest in microbiology further, she worked in biotech for a little bit, and then she came to us to start her PhD. I meant to show you nice photos of Julia while I was saying that. There we go, there's Julia, doing work of various kinds. So she joined the lab in early 2015 after doing her MCEB rotations, and shortly after that, we learned that my NSF proposal to go to the deep sea hydrothermal chimney system to Lost City was funded, and at the time we thought that was perfect timing. There would be just enough time for Julia to prepare and design some cool experiments that would be the foundation of her PhD. And then for various bureaucratic and scheduling and logistics reasons, the expedition was delayed. And then it was delayed again, and then it was delayed again. And so instead of going to the seafloor to study hydrothermal chimneys, Julia went to our computer server and did a whole PhD working with old data and samples that we had archived from previous expeditions. And that will be the bulk of what she's going to tell you about today. She did a whole PhD worth of work with older data and samples than we had been planning for her to work on. However, Julia really likes to spend time outside. So in addition to that PhD, she also did another PhD on the Bonneville Salt Flats. This is a quote unquote side project in collaboration with Brenda Bowen that was a whole PhD worth of work on its own. And she's just going to tell you a little bit about that today. But I want to mention that there are more experiments and metagenomic data for this project that Julia is not going to talk about, but that we're all still excited to carry forward. And because Julia is such a great team member, she did in fact finally join us on the expedition when it finally sailed in late 2018. And she was an absolutely critical member of our team and did some of the most important experiments and collected critical samples for the whole project, even though we knew that those data and samples probably wouldn't make it into her dissertation. But the story has a happy ending because Julia was recently awarded a NASA astrobiology postdoctoral fellowship to work with my friend and colleague Beth Orkett at the Bigelow Laboratory for Ocean Sciences, where she'll get to continue working on those samples that she helped to collect while learning new techniques and approaches at Bigelow with Beth. So we're all very excited that she will get to continue working on the project, and I'm excited to work with Julia as a colleague and a collaborator. Finally, I want to mention Julia has been extremely active in scientific outreach and communication in addition to all the research projects that she's been doing. Here she is talking about extremophiles at the Green Pig for Science on Tap. She's also given presentations at the Natural History Museum. And last summer, she organized and hosted an international astrobiology conference only for graduate students that attracted about 70 students from all around the world. All right, before I hand it over to Julia, a few ground rules. You will all be muted during the presentation, so type your questions into the chat window. If it seems like it's a good question to interrupt Julia with, I will do so, I'll moderate. Afterwards, there will be a Q&A period. Again, type your questions into the chat window and I'll read them out loud. After that, the committee will meet with Julia privately, and then assuming all goes well, and I'm sure it will, join us again at a different Zoom link, which is here and will be emailed out by Shannon. And we're guessing around 4 o'clock today for the Zoom celebration. All right, I will stop sharing and Julia can start sharing, and I am going to unmute you all so we can all welcome Julia via Zoom.
Hi, everybody. Okay. Okay, can you guys, I think you should be able to hear me now. Okay, so I'm gonna share my screen, and hopefully once I share it, just let me know if you can see my slides verbally, because I won't be able to see your thumbs up or anything. So let's do this. Okay, so that should be sharing. Can everybody see the slides okay?
Look good.
Okay. Okay, great. So, Billy, thank you for that wonderful introduction. That was very kind of you. It highlighted a lot of the cool work that I've been able to do on both of these environments over the last 6 years, the Bonneville Salt Flats and the Lost City. And we can link these two environments together by the phrase endolithic environments. So what is endolith? It's really just a fancy word for a microbe that lives with rocks. So the rocks are providing some amount of protection and against these things like environmental extremes like UV radiation and sunlight. And the microbes that are living with these rocks can either be living on the outside of these rocks in a biofilm or inside of the mineral pore spaces in the rock, or actually within the crystalline mineral structure itself. These things are really important because they're often involved in these crucial biogeochemical cycles that are providing macronutrients to the rest of the ecosystem. And they're super common. There's endolithic environments outside of your window, in the rocks and the soils, in the deep subsurface, even on monuments that we build. So really, endoliths are kind of everywhere. So the two environments, like I said, that we study are the Bonneville Salt Flats, which is located in northwestern Utah. For those of you that have been out there, you've probably seen what a cool environment it is. And I know some of you have been to the Lost City on here, some of the collaborators that have joined me today. But for those of you who haven't been there, it's a site that's located along the Mid-Atlantic Ridge in the middle of the Atlantic Ocean. So the questions that we're asking for both of these environments are seemingly very basic and very similar. We want to understand who is there, what does the microbial community look like, and then we want to understand what are they doing, what species are responsible for fixing the primary carbon for the ecosystem, how are they passing that around with other macronutrients, what other metabolisms are present. So these kind of basic who is there and what are they doing questions are what we're really interested in for these extreme environments. So I'm going to start by talking about the Bonneville Salt Flats, and this is what the area looks like for those of you who are joining us today that haven't been there. When you step out into this landscape, it just looks like it's barren and desolate for miles. However, it's actually teeming with life underneath the salt. And that is what I study and we'll talk about in a little bit. But first, I just kind of want to give you an idea of where it came from. So the Bonneville Salt Flats, as well as the Great Salt Lake, are remnants of this ancient Pleistocene Lake Bonneville that spilled out into Idaho and Oregon and left behind the salt flats as well as the Great Salt Lake. So the salt flats are valued both economically for the potash that's mined and the brines that are underneath the salts, and recreationally, especially to the racing community who uses it as an area to create these high-speed racing records. So the salt flats, a lot of people go out there for this annual racing event known as Speed Week. It's held yearly, and in 2014 and 2015, this event was canceled, and that caused a lot of raise and concern about what was going on out there. Why is this salt seemingly shrinking? And it sort of created this push to understand all of these processes that are happening out there. So Brenda Bowen at the University of Utah in the geology department decided that it would be a fantastic idea to get together all of these scientists that study different things, the hydrology, the geology, the biology, with some of the softer science people, because there is so much human interaction that's going on out there. So we're just a tiny piece of this large study that's really trying to connect these physical processes to these human socio-processes. So in 2016, I went out with some of Brenda Bowen's team, And we went out to the salt and we sampled from it as sterilely as possible and dug down into the salt to get our microbiology samples. We sampled at these different 8 locations, and we took samples according to these visual striations that we saw as we dug down into the salt. So you can see in these 8 pits, there's kind of these different visual layers that form that we tried to collect our samples by. And then half of that sample went to Jeremiah and Brenda Bowen's lab for geochemical analysis, and the other half of that went to me for DNA extraction and sequencing. So I think it's important to keep in mind here that when we extract the DNA from these environmental samples, we're getting all of the DNA that's in that sample. There's no way to isolate for organisms. We don't culture anything. So everything that I talk about that's DNA-based is really just the DNA that comes from every single organism in these environmental samples. And so the sequencing methods that we use, the 16S rRNA gene sequencing, I'm going to explain using an analogy of puzzles. So if you think of our environmental sample as containing all of these different little microbes, you can think of each one of these little microbes as being an individual puzzle. And all of the puzzle pieces within that puzzle are all of the genes that are within that microorganism. And so 16S rRNA gene sequencing is basically as if we were to pull out one single puzzle piece or one single gene from every single organism. So we're only sequencing that one gene from everything. And the gene that we're using is the 16S rRNA gene, and that's because this is a highly conserved protein and has been very clearly defined as a universal taxonomic marker for all domains of life. So we, in the Brazelton lab, are really interested in the bacteria and the archaea, so we sequence specifically for those. So 16S rRNA sequencing is using this 16S rRNA gene as a taxonomic barcode to answer the question, who is there? Okay, so that was the type of sequencing that I did at the salt flats that I'm going to talk to you about today. Jeremiah, and this is the really great thing about collaboration, is he was able to take the elemental and geological data from the samples that I gave him and sort them out into these 4 different groups. So you'll notice that there's this group 1, which consists mainly of the upper salts, and then this group 2, which is mainly just that sediment that's right under the salts, and then you have these other 2 groups that tend to be, depending on the site, in different spots. So what I was really interested in is which microbes are specifically enriched in each one of these different sediment categories. So I did differential abundance comparisons between the surface halites and the upper gypsum and the lower halites, and this 4th category, the lower halite and gypsum. And this is the results of that, which unless you're a microbiologist, you're probably not used to looking at these plots, and they look pretty crazy and there's a lot going on in there. But essentially what you're looking at is each one of these colors represents a specific taxonomy. And the amount of space that this color takes up is the amount of that enriched taxa that's present in this sample. So I got the enriched taxa from this comparison, and what was evident to me when I looked at all of this data is that in all of the comparisons with this group 2, or this upper gypsum layer that's just under the salt, is that there's lots more colors in this plot than some of the other comparisons. So another way of saying that is that there's a little bit, there's more different types of taxa that are enriched in this group 2 when compared to any of these other sediment groups. So that's really where the microbes are really liking to hang out in the salt flats. That's where they're most ported, is where you see this greater diversity of enriched taxa. So all of this is published. Our paper was accepted last year and came out called Robust Archaeal and Bacterial Communities Inhabit Shallow Subsurface Sediments of the Bonneville Salt Flats. And basically the main conclusions of this paper were that there is lots of life at the Bonneville Salt Flats. Even though it looks like a really barren, desolate area, there's tons of life. These are in no way limited in life. We got tons of DNA out of the samples, and we're able to characterize it pretty well. This upper gypsum layer just under the salt crust is where that life really likes to live. And we did some digging into the research to look at how the types of taxa that we find at the salt flats compared to these other salt flats that have been characterized around the globe, and found that it's very similar. So that kind of supports this old Bass-Becking adage that everything is everywhere and the environment just selects for it. So salty environments are selecting for similar types of organisms around the globe. Okay, so I'm going to transition now away from my salt flats work and into my work at the Lost City for the remainder of this talk. So just to remind you, the Lost City is our site in the Atlantic Ocean. So the Lost City sits right on this underwater mountain at about 900 meters down. The mountain's called the Atlantis Massif, and it's located at this really cool 90-degree bend on the Atlantic Ridge, which ends up popping up the mantle carbon that makes up the Atlantis Massif and drives the whole system. So I have this really beautiful video that we took in the 2018 expedition of what the fluids venting out of the chimneys look like. So you can see this kind of like warm, clear, shimmering fluid coming out. This is the end member hydrothermal fluid coming out of the chimney, and you'll notice that it's not these big billowing black smokes that you kind of typically associate with hydrothermal systems. And that's because the entire system at the Lost City is driven by serpentinization reactions rather than magmatic activity. So serpentinization is essentially a geochemical reaction where the mantle or the rock that makes up this Atlantis Massif reacts with water and creates the minerals serpentinite and magnetite. And this process releases heat, so it increases the heat of the fluids that are coming out, and it also increases the pH of the fluids to around 9 or 11. And this increase in pH is causing the calcium carbonate in the seawater to precipitate out and form these beautiful chimney structures. The process is also creating these very biologically relevant chemicals, hydrogen sulfide, hydrogen, and methane. And this is really important because this is really what is fueling all of the life that we see growing all over these chimneys. This is one of the few environments on Earth that is surviving and existing all from this reaction and not sunlight. So this entire system, this entire ecosystem is not being fueled by sunlight. It's being fueled by serpentinization. And this is important if you think about how we're going to find life on other planets, because we know that similar active serpentinization is happening in these icy moons of Jupiter and Saturn, Europa and Enceladus. And if we really want to understand what life would look like in these other planetary bodies, we really need to understand what it's doing here on Earth. So we're characterizing these microbial communities that we see growing on these gunky, snotty biofilms that are growing all over the Lost City chimneys. And previous studies have done a really good job at answering the question, who is there, using 16S rRNA sequencing and fish microscopy. And essentially the results of those studies showed that there are two very different types of biofilms that are grown on these chimneys. There's the chimney exterior biofilms that are comprised of very diverse bacteria. And then there's these interior biofilms that are made up mostly of this single species archaea, the LCMS, or a methanogen. So if you hear me talking about the Lost City archaea, Methanosarcinelles, LCMS, or methanogen, I'm talking about this one species that's growing inside of the chimneys. So we can kind of summarize all of the past data and sort of the environmental characteristics of these two different biofilms by saying that the biofilms on the chimney exterior are dominated by bacteria that are likely using sulfur compounds and methane. They're existing in this aerobic environment where they're getting access to both the serpentinite-derived fluids and the constituents in the seawater. So they have access to things like oxygen that are in the seawater, whereas the chimney interiors are much more extreme. They're continually bathed in those end member fluids, so they're much hotter. There's a higher pH. They probably don't have access to anything in the seawater, so that's a more anaerobic environment. And we don't see a lot of things growing there. They're mostly dominated by these biofilms containing the methanogen. So we're very interested in the carbon story at the Lost City. So we really want to know what are the primary carbon sources there. And that's largely because these serpentinite-derived fluids don't really contain a lot of CO2 in them. It's mostly precipitated out in that very alkaline environment. So these are the measurements for the CO2 or the dissolved DIC that's available in the fluids. And if Susan's on here, I'm sorry, I'm gonna very briefly talk about these things and kind of run through this data really fast. But essentially what the chemistry is saying is that we don't really have a lot of DIC in the fluids that are available to the microbial communities, but there is tons of methane and there's some formate and some lesser extent acetate that's available in them. So by concentration alone, the best sources for carbon into these microbial communities are either methane, formate, or seawater carbon sources. So just to kind of conclude this all, the chimney interiors likely don't have access to anything in the seawater, but formate and methane are available in really high concentrations. And then a lot of Susan Lang's work in the University of South Carolina has showed us that these formate and methane in the fluids is derived from this ancient mantle carbon source through these serpentinization reactions. So I'm going to even more briefly summarize the isotopic data. And essentially there's this interesting trait that you see between the seawater carbon and the fluid carbon, and that's that the seawater carbon for the delta 13C value is very positive, whereas all the fluid carbon sources have very negative values. And delta 13C is basically just a ratio of 12C versus 13C. So if we want to know which carbon source the microbes are using, we can look at what the isotopic signature for the carbon that's in microbial membranes looks like. And when we do that, we can see that these bacterial membranes, or that the carbon isolated from these samples that is from components in bacterial membranes is all negative, and the carbon isolated from things that are in specific to archaeal membranes has a mixed signal, but it's trending towards negative. And in cultured species, this sort of mixed, slightly positive signal has been linked to methanogens that are living under carbon limitations. So quickly to summarize all of that, these delta 13C values are suggesting that the microbial community is definitely using a mantle-derived carbon source, which is likely formate or methane, and that there is carbon limitation happening in this environment. Furthermore, we think that formate is super important because the values for the archaea, the methanogens, are not that consistent with when compared to values from cultured methanogens that use acetate as a starting point for methanogenesis, or the anaerobic oxidation of methane. And so keep in mind that these methanogens are living in an anaerobic environment, so the only way for them to use methane is this anaerobic oxidation of methane. So we can also look at the isotopic signature of the 14C, and when we do that, we can see that the seawater CO2 has tons of 14C in it, and that the 14C in the carbon from the fluids is depleted if it's methane, and a little bit very low in formate. The 14C signature from the carbon in the bacterial membranes and the archaeal membranes really does match the formate more than anything else, and is suggestive that they're not really reliant on the seawater CO2 source. So the 14C values from the microbial membranes are also indicating that these microbes are really reliant on some source of mantle-derived carbon. Again, that's either formate or methane. However, the delta 13C values aren't really consistent with methane, and our previous 16S studies that I briefly mentioned to you have found none of these organisms that are known to do this anaerobic oxidation of methane. So I hopefully I didn't butcher the isotopic stuff too much, but if you want to read really more about that, you can. I'd reference you to Susan's paper that was published in 2018 in Scientific Reports, deeply sourced formate fuel sulfate reducers, but not methanogens at the Lost City hydrothermal field. And so you'll notice that there's things about sulfate reducers using the formate and not methanogens. This is a little bit of the genomic work that I've done to try to tease out who is this formate user, because the geochemistry tells us that formate is really important for the microbial system. However, it can't really tell us who is that formate user. We really need to use genomics for that. So I'm not going to talk too much about this genomic work that I did in this paper, because since then we've done some new stuff that's in line with this, but a little bit more interesting. So a lot of my Lost City project was really focused on identifying who this formate user is and how they're supplying carbon to the rest of the ecosystem. Are they oxidizing formate into CO2 that's briefly available and leaking out of the cell? Are they providing carbon to the rest of it through biomass? How is this formate user really passing carbon along to the ecosystem, and who is this formate user? So with 16S RNA gene sequencing, I think I've shown so far that you can answer the question, who is there? But if we want to answer the question, what are they doing, or more specifically, who is doing what, to identify who is using the formate, we need to turn to metagenomic sequencing. So if we go back to this puzzle analogy, with 16S, we were sequencing only one gene from everything. With metagenomic sequencing, we sequence everything. So we're taking all of the puzzle pieces from all of the puzzles and looking at them, or all of the genes from all of the organisms and looking at them. And it really is as if you take 100 puzzles and just dump all their puzzle pieces out on the ground, mix them up. And so if you want to go backwards, so if you have one gene of interest, or like a formate utilization gene, and you want to go backwards and find out what puzzle does this piece belong into, it's a little bit of a computational nightmare. So before I get into how we actually do that, I'm going to talk very briefly about the formate utilization genes that this project was focused on. So the first gene is this formate dehydrogenase, which is responsible for catalyzing the bidirectional oxidation of formate to CO2, and then I can hand off some electrons to something like NADH or ferredoxin. The other two really important genes for a formate utilizing organism is a formate transporter. So formate doesn't just passively diffuse into the cell, it has to actively be brought in by something. And so there are two known formate transporters, the FTHC and the FOCA. So now I'm going to tell you a little bit about how we go backwards and figure out who's doing what with metagenomics. So we can use a process called binning in order to do that. So what binning does is it takes every chunk of DNA or contig that was assembled from these short reads that we're sequencing, and it can take the tetranucleotide frequency of that contig, which is essentially just the frequency in which you see these combos of 4 nucleotides, so like AACG, ATAT. So how often do you see this tetranucleotide frequency signatures in combination with the read coverage, and sort it out into things that are similar. So the binning algorithm that I used for this project is called AlbaWaka. And at the end of this, you get something called a bin, which also represents a metagenomically assembled genome. So obviously you're just relying on this algorithm, and you need a way to check the quality of that bin after you obtain this bin. So in our lab, we use something called CheckM, which uses data of single copy marker genes to assess the percent completeness and the percent contamination of each one of these bins. So essentially, for all cultured organisms, there are a certain number of these single copy genes that they should have. And if they have all of them, then you have a 100% complete, it's very complete bin. However, if you have multiple copies of them, then you have contamination, because they should only have one of these genes. So what CheckM does is it looks at your bin, and it looks at how many of these single copy marker genes you have in that, and then spits out a percent completion and a percent contamination. So if you have your bin that contains your gene of interest, like the formate dehydrogenase, and you have a really low percent completion or really high percent contamination, what do you do then? Well, that's exactly what happened to me. After running Abawaka, I had this starting bin that contained a formate dehydrogenase that was 100% complete with close to 400% contamination. Obviously, this is super, super high percent contamination. Can't really use this to do much. Nobody will really trust you if you say, oh, my bin was 400% contaminated, so you have to refine it. You have to chop some things out of there. So how do you decide what to remove from this bin? Well, I decided to start by using taxonomy. So we have a program, Phylopithia, that I used to assign taxonomy to each one of these contigs or chunks of DNA in my bin. And then I found what the taxonomy was for the contig that contained my gene of interest. So in this case, the formate dehydrogenase gene was on a contig that was identified as sulforovum by Phylopithia. And then I sorted out my bin and removed basically everything that was not a sulforovum. So I'm calling this refining by contig taxonomy. So after taking this bin and refining it by the contig taxonomy, I was able to get something that was 96% complete and 52% contaminated. So this didn't give me a huge reduction in completeness, which is good, but it did give me a massive reduction in contamination, which is also good, but 52% is still a pretty high number. So to get that down further, I moved to a separate program called Anvio, which gives you this visual clustering of your contigs by similarity. So when I did that with this bin, you can notice right away that there's these two very distinct groups, the one highlighted in green and the one that's gray. So I split the bin in half by these two groups and removed one of them and then kept the chunk that had my contig of interest in it. So after refining my bin with Anvio, I ended up with something that was actually really, really good and able to work with. So it didn't remove any of the completeness, but it did remove a ton of the contamination. So that's how I obtained all of the bins that I'm going to talk to you throughout the rest of this talk. So the first bin is the sulforovum bin. This is the one that I just told you about how I refined. And the fact that it's sulforovum is really good because this is something that we know is very common in hydrothermal chimneys. So the organism checks out for what we would expect to find at the lost city. The second one is this chloroflexi bin that's 70% complete with 4.73% contamination. And this is also really good because chloroflexes are common in hot springs, and they've recently been described in a number of increasing subsurface samples. And then the last bin that we're going to talk about today is the LCMS, or this methanogen that's making up that interior biofilm. And an interesting thing about this organism is that it has only been described in 2 places across the globe, the Lost City and this other serpentinite hosted system, the Prony Bay. The Prony Bay is very different from the Lost City because it also has geothermal activity in that system. So it's not quite driven solely by serpentinization like the Lost City, but there is an element of serpentinization happening there. So we're going to go through each one of these 3 bins that I just mentioned, and I'll tell you how I use genomics to identify which carbon sources they're using. We'll talk a little bit about whether their central carbon metabolisms are suggesting if the organism can grow autotrophically or heterotrophically, so it makes its own food or it needs to scavenge organic carbon from its environment. And then we'll talk a little bit about the bioenergetics of each one of these populations. And then we'll try to put everything into a bit of a better environmental context, sort of say, why does it matter? Who cares that it has this list of genes? What is it actually doing in the environment? So we're trying to really build up, by using genomics, this larger picture of what's happening in the Lost City ecosystem. How is the carbon being cycled around? Who's using it? What parts of the chimney do they live in? Are they on these chimney interiors, chimney exteriors? And how are they passing things to each other?
Hey, Julia.
So we'll start.
Can I interrupt you?
Yeah.
Martin had a question I think that would be good to clarify right now. Does contamination mean environmental contamination or genomic heterogeneity?
Yeah, that's a really good question. So contamination means it means that there's more of these single marker copy genes. So it would be more genomic heterogeneity. Like, so you don't actually have a single bin that's that one organism, you have some DNA from something else that's contaminating it. So not environmental contamination per se, like something from your hands or something.
It's a mixture of multiple genomes.
Yeah, a mixture of multiple genomes, yeah.
Great, thanks.
Okay. Okay, so we'll continue on. So I mentioned to you guys that I had this contig of interest that drove my interest in this bin. This is what that contig actually looks like. So here is the formate transporter gene on that, and then you'll notice downstream of it, you have these hydrogenase 4 multiple genes in this operon, and then the formate dehydrogenase. So the products of these genes work together to form a large formate oxidizing complex called the formate hydrogen lyase. And what's interesting to note is in these hydrogenase 4 genes, there's multiple different types, and this hydrogenase 4 is the type that's known to be unaffected by high pH. So this is cool for the lost city because the lost city is a really high pH environment, so you would expect genes to be adapted to still work in these really alkophilic places, and I could see that directly on this contig that was suggesting formate utilization. So we can take all of the information that's in the bin and sort of build this entire picture of how the cell works. And there's kind of a lot going on in this figure, but we'll walk through it one by one. So here's the formate utilization complexes. You have the formate transporter and then the FHL that's oxidizing the formate into CO2. And then you also have evidence in this bin for ACS, which is indicating that the organism can bring in acetate from the environment, convert it to acetyl-CoA, which is a central point in a lot of other different metabolic pathways. So the carbon sources for the sulfur-driven population are formate, acetate, and CO2. But what about whether this thing's autotrophic or heterotrophic? So it can actually fix the carbon from formate by using this reverse TCA cycle. So the reverse TCA cycle, a lot of you are familiar with the forward TCA cycle, but you probably don't know about the reverse TCA cycle because it's pretty specific to microbes. But it basically has a replacement of these 4 different enzymes. Instead of succinate dehydrogenase, fumarate reductase is used. Instead of citrate synthase, the ATP citrate lyase is used. There's a pyruvate. There's the PFOR there. There's the 2-oxoglutarate pyridoxine reductase. So it's using the TCA cycle essentially in reverse with the modification of these enzymes in order to fix carbon instead of break down carbon. What about the forward direction? So it's unclear just based on genomics alone whether it can also run this cycle in the forward direction. It does have the succinate dehydrogenase to replace the fumarate reductase, and it's likely the other two, or the oxoglutarate pyridoxine reductase and the PFOR, can run in the opposite directions, but the ATP citrate lyase is unclear. There have been studies done with cultured sulfidobacillar species that have shown that this enzyme can be bidirectional and doesn't need to be replaced with the citrate synthase. However, you'd have to actually culture this organism and grow it in order to find that out for this population. So it's definitely autotrophic and it's possibly heterotrophic, in addition to possibly using the full forward TCA cycle. It's possible that it uses a shunted version of the TCA cycle to break down things like amino acids or something. And I did find evidence for amino acid transporters in the bin also. Okay, so let's shift away from carbon for a minute and talk about the bioenergetics of the cell. So there's a couple different places where the cell can generate reducing power from. One is that formate dehydrogenase, the other are these membrane-bound hydrogenases. This is great because there's lots of hydrogen in the system. Hydrogen is a really great source of reducing power. And then it's feeding this reducing power into things like the electron transport chain and things like the reverse TCA cycle, because instead of generating energy, the reverse TCA cycle is consuming energy. And then this bin also contained the sulfide quinone oxidoreductase, which is donating electrons into the electron transport chain from hydrogen sulfides. And this is something that's fairly common to sulforobums. So through the genomic evidence, we can identify the electron donors as formate hydrogen and hydrogen sulfide. And then what I found really interesting about this genome is that it actually has 3 different terminal electron acceptors in the electron transport chain. So it has this fumate reductase, which we talked about, which is part of the reverse TCA cycle. But it also has the complex 3 and 4 that aerobic organisms use to use oxygen as a terminal electron acceptor, in addition to a nitrate reductase. So it has a very flexible metabolism and a lot of these different electron acceptors that it can use depending on cues that it's receiving from the environment. So it's unlikely that it's actually making all of these things at the same time. It's likely getting environmental cues and deciding which one of these things to use. So it's going to get the most energy from oxygen if it's available, but if oxygen's not available, that it can grow anaerobically using nitrates. So we're kind of talking a little bit about the environmental context of the genes, and even just by this picture alone, we can get a good idea of things in the environment that are really important for its growth. Formate, hydrogen, hydrogen sulfide, oxygen, and nitrates. So we can kind of summarize all of these things for the sulforobum population by saying that it has a very flexible metabolism. It can either grow aerobically or anaerobically. Another interesting thing that I haven't mentioned yet is that there were genes for these transporters that excrete these capsular polysaccharides, which are involved in biofilm formation. So it has a role in forming these biofilms that we see growing on the chimneys. And then from those amino acid transporters, it can scavenge amino acids from the environment. So it's probably living somewhere near other cells, and it's using constituents from the chimneys like nitrates, hydrogen sulfide, hydrogen formate. So it definitely has access to the fluids, but also has access to the seawater because it can use oxygen. So this organism is likely adapted to a transition zone that lies somewhere between the interior and exterior of the chimneys. Okay, so we're going to switch to talking about the chloroflexi bin now. And so just to remind you, this bin was 70% complete with around 5% contamination. So this was the contig in the chloroflexi bin that got me interested in it. This FOCA right here is the formate transporter, and directly adjacent to that was a formate dehydrogenase gene. So again, you have on the same piece of DNA evidence for formate utilization. And so just like the sulforobum bin, we're going to kind of go through and identify the same things. So to show you a similar cell picture, but for the chloroflexi cell, we have here the entry point for formate, the FOCA, and then the formate dehydrogenase. So it's able to utilize formate from the environment, but also has the same ACS that sulforobum does that allows it to use acetate from the environment. So the carbon sources, according to the genomics, are formate, acetate, and carbon dioxide. What about whether it's autotrophic or heterotrophic? So it is able to, just like the sulforobum, fix that CO2 from formate, but it's doing it through a different pathway. So the chloroflexi is using this reductive pentose phosphate cycle, which is similar to what plants use. Those of you who are listening that are familiar with plant biology probably have heard of rubisco before. Rubisco is the enzyme that fixes carbon for plants, and this chloroflexi organism contains the same thing. What's interesting to note about the reductive pentose phosphate cycle is there were additional genes that indicated that it is using a carboxysome, which is a structure that isolates the rubisco enzyme to sort of concentrate the CO2 around it. So this is a structure that's present in really low CO2 environments. So again, this fits into our sort of natural ideas of what's happening in the lost city, this low CO2 hot environment. So something that was at first really interesting and confusing to me is that there's actually 2 carbon fixation pathways that are contained in this bin. You have the reductive pentose phosphate cycle that I just talked about, as well as the Woodland Hall pathway. So the Woodland Hall pathway is a pathway that organisms called acetogens use to fix carbon and make acetate. What's really interesting about the Woodland Hall pathway in this bin is that it's very incomplete and missing some key genes. And similar incomplete Woodland Hall pathways have been described in chloroflexy organism Dihalocucordes McCartyi that's been found in some of our single cell studies at the Lost City. And there was a paper that came out in 2014 that described that this Dihalocucordes McCartyi that they were able to actually culture and grow was using the Woodland Hall pathway backwards to make methionine rather than in the more forward direction to fix carbon. So it doesn't really make sense that you would have an organism with 2 carbon fixation pathways. So it's much more likely, especially considering the lack of other methionine biosynthesis pathways, that the chloroflexy is fixing carbon through the reductive pentose phosphate cycle and making methionine through this partial Woodland Hall pathway. So what about what it's doing with the fixed carbon? So it's able to fix the carbon through the reductive pentose phosphate cycle as glucose, like an autotroph, but it also contains this forward TCA cycle. So it's able to either break down its own glucose reserves or scavenge carbon from the environment. There were multiple sugar transporters inside of this genome, so it can scavenge organic carbon and grow heterotrophically, or it can grow autotrophically by fixing carbon. Okay, so we're gonna switch to the bioenergetics of the cell. So just like the sulforovum, it's likely that this thing is very reliant on hydrogen in the environment. Hydrogen's super, super abundant in the Lost City, so this isn't surprising at all. It's a great source of reducing power. It can also get reducing power from the formate dehydrogenase and the forward TCA cycle. And then it's, again, using that reducing power that's generated to go into energetically demanding metabolisms like fixing carbon, or it's feeding it into the electron transport chain. So the electron donors are formate organic carbon if it's growing heterotrophically, and hydrogen. So I want to talk about this one complex right here. So complex 1 is something that aerobic organisms have. Also, it's the NADH dehydrogenase, and so it's normally pumps through 4 hydrogens, but if you were super aware, you could see a 5 down here. So I'm going to talk about why that's a 5 and not a 4. So there was a paper that came out very recently that described these interesting nuo complexes that have incorporated an additional M subunit into the structure. So the complex 1 or nuo complex is one of these massive enzymes that has a lot of different subunits. And with this additional M2 subunit incorporated into it, it's able to actually pump an additional proton across the membrane. So the same paper, Chadwick et al. Described all of these different complex, interesting clade according to phylogenetic relationships and stuff. So not only do they cluster out according to the taxonomy of the species containing them, but also whether they're this M1 type or whether they're this nuo M1 type or nuo M2 type. So using all of his sequences as a reference, I built a tree with all of my nuo M genes that were in this bin and found that they clustered into these 3 clades. So this was super interesting because in particular, this clade 1 right here is the chloroflexy clade, and I had 2 genes that were right next to each other that were clustering with the references for nuo M1 and nuo M2. So I use this as a line of support that the chloroflexy also has this modified nuo complex. In addition, the gene order matched what was described by Chadwick et al. For chloroflexy species. Both of those 2 adjacent nuo M genes were flanked by an L and an N, as well as in the genome on a separate piece of DNA. There was a new B, CD fusion, and I. So he described this as very specific genomic features for chloroflexes that had this NOM2 complex. So that is why we believe that this chloroflexi population has something similar and is able to generate additional proton pumping force per reaction cycle. So this is something that is probably very useful for either slow growing environments or as an adaptation to an alkophilic environment where this proton motive force might be harder to generate and sustain. So the electron acceptors that are present in this genome is really just one, the polysulfide reductase, which is using polysulfides as the terminal electron acceptor. But you also notice that there's a complex 4 here. So complex 4 is what aerobic organisms use in order to complex 4 is what aerobic organisms use that is accepting the oxygen. However, it's kind of a weird thing to see in this bin because there was a number of other anaerobic enzymes that were present. So there's these 4 genes, the COTH ACS complex, which is super sensitive to oxygen. There's also this aldehyde ferredoxin oxidoreductase. And then there's anaerobic forms of these 2 genes. But maybe more perplexing, there's a lack of the complex 3 genes, which is what is donating the reduced cytochrome to complex 4 in aerobic organisms. So if it is actually making this complex 4, where's the reduced cytochrome coming from? This is very confusing. Perhaps it's coming from something like the HDR, which I did find in this genome. It's been proposed that this is a source of reduced cytochrome in things like methanogens before. So it's possible that the HDR is providing the reduced cytochrome to complex 4, and that the whole entire role of this thing, rather than serving as a terminal electron acceptor, is just to remove oxygen that's poisonous to the organism. And this is a genomic feature that's been described in other anaerobic chloroflexes, is that they have these things in them, but they're probably just there to remove whatever oxygen filters in. Okay, so the electron donors for the chloroflexi population are formate, organic carbon, and hydrogen, whereas the electron acceptor is these polysulfides. So what about the environmental context of all of this? So you can kind of see right away polysulfides, which we do know exist in the fluids to some level, are probably pretty important for it. Hydrogen, organic carbon, this thing is probably growing as a heterotroph more often than as an autotroph, but it can grow autotrophically, it has to. And then it's using formate also. So it's very interesting that our best candidate for an anaerobic formate user is probably also a heterotroph. So I want to talk about some other genomic features that are pretty interesting. So I told you why I think this thing is likely anaerobic, but I kind of want to explain to you why I think it's also thermophilic. So this is something that is super hard to detect in genomes, and it's a really difficult thing to get at. You would think that it would be more obvious, but it's really not. So that carbon fixation pathway that this thing has, the reductive pentose phosphate cycle, is missing only one enzyme. And that is the enzyme that is responsible for producing G3P. And G3P is a product that's just really not that stable in heat. This enzyme has been shown to be modified in a lot of other thermophilic organisms. In my chloroflexi bin, it was completely missing. So that could be, maybe there's some novel form of it that our annotations or that our database just didn't match up to it because it's a novel form. In addition, there's this other enzyme that is often modified in thermophilic organisms. So this is an enzyme that's involved in gluconeogenesis, or the formation of glucose. And when I did a little bit of work to see which organisms this thing was most similar to, it came out most similar to this other thermophilic organism. So kind of all of that puts this chloroflexi in this context where it's living in a likely really low CO2 environment. It's probably liking to live heterotropically because that's a really good way to grow for a microbe. So it's probably living near other cells. I went over all of our evidence for why I think this is an anaerobic organism, and then we just talked about why I think it's a thermophile. And then it has this cool modified version of a complex 1 that indicates that it's slow growing or adapted to a more alkaline environment. So all of this kind of puts this thing as living in those interior chimneys, probably right next to the LCMS. It's very interesting that it's a really good candidate for a formate user for the LCMS, because a lot of our other work has shown that LCMS is definitely not able to use formate, which is the most abundant and the most likely carbon source for it. So it's very possible that this LCMS is super, super reliant on something like the chloroflexi to live. So we'll kind of briefly talk about the LCMS bin in the time that's remaining here. So like I said, it cannot use formate. We were able to obtain a pretty good bin for it, and there's still no evidence of formate utilization. So that's cool. So what is it using instead? Well, it has the genes that allow it to do acetoclastic methanogenesis. It's probably using CO2 for methanogenesis, and then it can also use these methanol or methylated compounds for methanogenesis. So the carbon sources are methanol or methylated compounds, acetate, and CO2. It's autotrophic because it's a methanogen. We don't really just get into that too much. And then the bioenergetics of the cell. So just like all of the other organisms that I've described, it's likely very reliant on hydrogen in the ecosystem. Again, this just isn't surprising. When you think about the velocity, you would predict that to happen. Hydrogen is a pretty good source of reducing power. So the electron donor is hydrogen, and then the electron acceptor, because it's a methanogen, is carbon. We won't really get into that too much. So the environmental context of all these genes is that it's not a formate user. It's living in an environment where it's reliant on the formate. It's all of the isotopic signatures in its membrane are suggesting that it's using formate, but it's not a formate user. It has no genes that are allowing it to use it, but it is using hydrogen. So it's likely using the CO2 that's briefly available leaked out from something like the chloroflexi, but it might just be doing something where it's giving nitrogen to the rest of the cells. So it's not just, so it could be more of a syntropic interaction rather than something like it's just stealing carbon from everything else. It could be playing this super important role for providing biologically available nitrogen to the rest of the community. It does contain all these NIF genes for the nitrogenase that's responsible for converting nitrogen gas into ammonia. So so basically there's no evidence for formate utilization, even with this more in-depth study that we've done. It's a strict anaerobe. It's capable of nitrogen gas fixation, which is super important ecologically. But it's not using formate. It's definitely reliant on something like the chloroflexi, which is likely living in these hot anaerobic interior chimneys. And then the sulforobum is likely living just on the outside of the chimneys. It could be passing CO2 to the LCMS through the chimneys or something like that. And then the LCMS is actually able to release bicarbonates that could help form the chimneys. So you kind of have this interesting picture where the LCMS and the chloroflexi are both working together to cycle carbon around. And if you're interested in a lot of that genetic stuff that I got into, we had a paper that was just accepted recently in AEM, Genomic Evidence for Formate Metabolism by Chloroflexi as a Key to Unlocking Deep Carbon in the Lost City Microbial Ecosystems. And I think it's out now. If it's not out now, you can see it pretty soon. So with that, I would like to go over my acknowledgements before I take questions. First and foremost, I really want to acknowledge Billy. He's been an absolutely wonderful advisor through all of this. I don't really know if I could have survived this process with the worst advisor. I want to just thank you for letting me kind of go off on all of my little tangents, and even when they were dead ends, just sort of independently explore whatever I found intellectually interesting. And I also want to thank you for letting me do things like the outreach and the conference organization. I think that really truly enriched my experience as a grad student here, and it gave me a greater sense of belonging within the scientific community in general. Sherry, you've been a really great lab mate. Really glad to share this time with you and all of the lab, really, including our former students. Emily was out there sampling with me at the salt flats and helped with a lot of the DNA extractions. Lizette does a lot of DNA extractions in our lab that's super helpful. Alex basically taught me bioinformatics and was incredibly patient. And I don't know if you're logged on, but if you are, thank you for that. And then something that I've really loved about all of this work is our collaborators. I feel like I've gotten to work with some of the most exceptional teams, both the Lost City team and Brenda's team on the Salt Flats. You guys have been really wonderful to work with. Like, just thank you for all of your patience with me. And I think that it's really cool that I do a science where I can work with people outside of my field and sort of get to know how to talk to them about their science and our science. And I think that that should be a more common thing for sure. And then I want to thank my committee, particularly for being super helpful with some of the metabolic pathway stuff. And then lastly, I know there's a number of my friends and family on here. Thank you guys for just everything and existing. Okay, and I'll take questions now.
265. Back up my.
Okay, so I have some questions in the chat window. Please type more questions into the chat window if you would like. If your question isn't typable, you can also try raising your hand, and I'll see if I notice that you are raising your hand. All right, Julia, first question. Can metagenome analyses be applied to detect viral loads in a human population, perhaps in sewage, for example? Oh, you're muted. Sorry, I muted you.
No, that's fine. I can unmute myself. Okay. Yes, so you can detect viral genes in a metagenome. I have never done any of this. We do have some collaborators that are doing this with some of our Lost City metagenomes. I think from that experience that it's much easier to do if you collect your sample, filtering out all of the cells, so you're removing a lot of the cellular DNA and trapping only the small particles and then sequencing that. So I don't know like how easy that is, I guess, is what I'm trying to say. But yeah, you theoretically can do something like that.
Yep. How about on Mars or Europa? What are the chances of doing some remote DNA sampling and sequencing extraterrestrially?
Yeah, so that obviously is a super, super good question, and one that I wish that I could say yes to, but as of right now, you still need humans involved. So unless we send humans to those planets, we can't do DNA sequencing and send the data back. There's still steps in the DNA extraction and the library prep that preps the DNA in order to sequence it, that you need a human hand to do these things. So there's definitely been some cool work with microfluidics where we're working on building some sort of technology that does all of these steps. But as far as I know, and I could be wrong, there could be a lab that's come out with something, but as far as I know, we're not able to do everything completely remotely. And the problem with having a human do it is that we don't have a way to collect samples and bring them back here yet. I think the problem with that is that fuel to do that, like the rocket fuel to do that, is just too heavy, and there's no way to put enough rocket fuel on something to send it there and back. And Billy might actually have a better answer for that than I do. But
I think that's a pretty good answer. Another question is from Evan Kipnis about the C14. So how does this high C14 concentration equate to low seawater usage? Is C14 fractionated during serpentinization or something else?
Yeah, so I am really bad with isotope chemistry. I'll just preempt this question by saying that. So I think that the 14C data and why we think that that's telling us that the seawater CO2 is not being used is more that the signature from the carbon in the membranes is matching more of these mantle-derived sources rather than the seawater source. So I actually have a slide. Hang on, I think I have to stop sharing this for a minute. I have a slide that gives you the actual numbers, which might be a bit more satisfying. Okay. Oh shoot. It's not doing it. Okay, can you guys see that?
Yep.
Okay, yeah. So this might be a little bit more satisfying. So if you can see, if I had thrown up the actual numbers, which I didn't for my talk, just for simplicity.
You have frozen, Julia, so whatever you're saying now, you might have to repeat.
The seawater is, whereas the 14C from, yeah. Okay. I think everything is frozen.
Okay, now you're back. Try a concise explanation one more time.
Okay. Okay. So the 14C of seawater is around 1 because they would have a much higher signature.
Yeah. So the biomass is C14 dead or mostly dead. Evan is suggesting that sharing the screen may be having a bandwidth issue.
Oh, okay, let's see.
But I think that probably answers the question. We can go ahead. Dave Blair has a question. I think that's not I think a good question for the general audience. Does the chloroflexy also have bicarbonate transporters? Because you mentioned the carboxysome. So do you think it's transporting bicarbonate for the carboxysome? Well, it's a good thing that Julia froze after her talk. Okay.
Ooh.
Oh, are you back? Okay.
Yeah, maybe. I don't know. Is that working?
Now you're, for now, but you're really fuzzy. So I think there's a bandwidth.
All right, I apologize. It must be my connection here is probably a little bad. I don't have a way to plug in directly with an Ethernet cable, so it'll just have to be what it is, I guess.
All right, let's do this last question. Bicarbonate. Do you think they're transporting bicarbonate?
Okay. I don't think I found any evidence for that. I could dig into it a little deeper and look for it, but from the work that I've done, I don't think so.
Okay.
They do have a carbonic anhydrase, so they are able to convert the CO2 to bicarbonate. That's maybe slightly unsatisfying, but.
And then Katrina Twing has a question about, do you know if the chloroflexy had been found in previous studies? I honestly don't really remember.
Yeah, so I'm assuming previous studies at the Lost City,
Yeah.
and I don't think that any of Billy's past work found chloroflexi, but I do know that Jackie in Beth Orcutt's lab, postdoc in Beth Orcutt's lab, has found chloroflexi in some of her work with the Atlantis Massif. In addition to Sherry with some of her work on the rock samples for the Atlantis Massif. That's the mountain that the Lost City sits on, for the rest of you guys that aren't Lost City collaborators. So we have found chloroflexi in those samples, as well as some of the new work that Beth has done with single cell sequencing in the new Lost City fluids that were collected. There are chloroflexi, and one of them is that Dihalococordes macardii. I guess on a tangent, we did briefly look at whether any of these specific Woodland Hall, this one particular gene that was missing from the Woodland Hall pathway was present in any of these, and it was also missing. So there's some very loose evidence that that's a yes.
Great. I think that's a good place to stop. Thank you, everybody, for attending virtually. I'll unmute you all again for one last round of applause. And
thank you all for making it too.
And around 4 o'clock-ish, we'll have a celebration on Zoom, different meeting ID. So see you then and there. Bye. Thank you.
Bye.